Welcome to the Microscopists, a bite-sized bio podcast, hosted by Peter O'Toole, sponsored by Zeiss Microscopy. Today on the Microscopists.
Today on the Microscopists, I'm joined by Mike Dustin of the Kennedy Institute of Rheumatology, and he talks about how he tweaked his grant applications that made all the difference to getting them funded.
And then we went from almost like that kind of triage point to getting like the top score in a grant round when it worked.
Where the convention for naming antibodies came from.
So one molecule might have 20 names based on different people making these antibodies. So they decided to get together all of the people who were making antibodies and analyzing these surface molecules, exchange the antibodies, and this started the CD thing, which some, CD1 through now they're into the hundreds.
And which is best, X, Twitter, or LinkedIn for networking and science communication?
Twitter, X type thing. I have an account and I'll tweet papers like, preprints and things. But basically most of the real utility comes from LinkedIn. Yeah.
All in this episode of the Microscopists. Hi, welcome to the Microscopists. I'm Peter O'Toole from the University of York, and today I'm joined by Michael Dustin from the University of Oxford. Mike, how are you today?
Fantastic. Thanks, Pete.
Actually, I should probably introduce you in a bit better as a groundbreaking, famous immunologist that we're now very lucky to have over here in the UK. And that's when you go red and get bad.
Of course. Yeah, no, it's been great. It's been great being here. I've been here 10 years now at the University of Oxford. And yeah, it's been fantastic. I've really been enjoying my, it's almost like our one of the things that enabled my wife and I to do this was our daughter getting to kind of college age and leaving home and things. And, so we had like this empty nest and kind of, but just decided, okay, we can be postdocs again and basically go off and do something totally different, which has been pretty much our last 10 years.
So your wife is also a scientist?
Yeah. So actually, we're both working at the University of Oxford. Previously we'd had, worked in different academic institutions in the same cities in St. Louis and New York. But in Oxford, basically, we were initially in different institute buildings. I was at the Kennedy Institute. She was at the Medawar, and now we actually both work at the Kennedy. So again, this is something we're both really kind of enjoying. I guess learning about Oxford and learning about doing research in a different setting. It's been very enjoyable.
And I say let's stay on this point then. You say you're enjoying Oxford, getting to know Oxford. How are you finding coming over to the UK culturally?
Yeah, like we work in English, but of course, a lot of the little sort of expressions and things and way people use language are subtly different. So I guess I've had to kind of recalibrate to understanding when I'm being told no indirectly or yes, I'm very excited about that, just trying to figure sort out sometimes like what kind of reaction I'm getting. But I think in general, like Oxford's been like a new playground kind of scientifically and also culturally. I'm affiliated with the college for the past couple of years, which has been a really interesting experience with Brasenose College. And so that's sort of, I'm in a research institute. So in a lot of ways, my research more or less has been, doing research in different exciting places. And this is, like a new exciting place to do research. It was, different colleagues, different specific opportunities and things. But, the teaching system in Oxford is really different than the teaching system I experienced, say, at Boston University as an undergrad. So I was in these, like huge lectures and these, kind of there were discussion sections, but it was really kind of not the kind of, tutorial system that Oxford and Cambridge use, where they're, really getting together with and kind of customizing that educational experience. So I haven't, done tutorials yet, but this is now something I'm getting, kind of, hopefully I will before, now that I'm not affiliated with the college and get a chance to, learn about this, different way of approaching, undergraduate education. And I think, and I'm really also a strong believer in, getting into labs early and having, kind of hands-on research experience at the undergraduate level, and I'm trying to sort of see if I can pitch this also to the folks at Brasenose, and to kind of maybe increase opportunities for that early experience, which maybe is again maybe considered difficult to fit into the curriculum in a in say Oxford's programs. So again, it's something that I'm kind of interested in, maybe seeing, although obviously this is a 1,000-year-old institution and things, I'm probably not going to have too much of an impact on how they do things, but at the same time, I'm interested in trying to tweak little things about how they might provide those experiences, which were so formative for me.
Well, it's so interesting you say that. Just last week, we interviewed for summer studentships at York. We got something called Generation Research, which Gillian Barlow is running at York. And the quality of the students, and actually it was the second year students really shone through, and they won't have had much lab experience. They'll have had some big lab experience from not, getting into it. And it was really, they were mind-blowingly good. And I think the summer studentships, it probably changes their career path, because they get to really understand research during that time. And I think it then boosts their third and fourth year, because they're now more motivated, got a better appreciation of it. Maybe find more of a passion and love for science. I've got to say, these students already seem to love it, but I do think, and the nice thing about Generation Research is they're kind of almost blind CVs. So you don't get to know who they are, and they're funded. So they can afford to do the studentships, which is really cool. But actually, we had one really cool student. Well, we had three really cool ones, and we had to flip a coin, pick one. And I think I've just found funding from somewhere else to get a second in. But do you do studentships, summer studentships in the labs?
The medical students have this program they call FHS program. So they do have a 2 months or so for research in their medical curriculum. Otherwise, it would vary quite a bit by department in terms of biochemistry has some kind of also some research opportunities and things. But the thing that I kind of really benefited from as an undergraduate was being able to kind of follow a project for like 2 years, and it wasn't obviously full time. It was really kind of quite part time, quite when it was possible to come into the lab and things. But it was it really I think that's really different than even like something where you have like a summer or something. So again, like that kind of thing where you can from like maybe second year bring together and again it might not be for everyone, but for people who really feel like they've gotten that bug for doing research, to have that opportunity to connect up these experiences as opposed to having them as multiple isolated internships or something. So again, it's but I recognizing that my experience may have been unique and worked out, but I know other people say when I was at Boston University, like other people were kind of doing that also, is kind of just getting started after maybe like I had cell biology as a course in second year, and then basically got really excited about cell biology, and then kind of just approached the PI and the professor from the course, and he basically said, "You want to do research in the lab, don't you?" And I was like, "I suppose, yeah." And he kind of then just captured me and kind of pulled me in and really helped me along. Really kind of a quite maybe something on the order of 5 to 10 hours a week or something. But and then with some interruptions, some breaks, but then otherwise letting me kind of go through a whole thing and actually write a paper at the end and stuff that we published in a peer-reviewed journal. And it was like it was an opportunity. So whether all that can be put into a program, or do you just sort of enable it? And I think in that case it was really kind of informally enabled. There was like a senior thesis project that was a course that basically I then fed into that already with quite a bit of work under my belt before that even thesis kind of senior thesis thing started.
It could just be a case of allowing it to happen
as well. I think that would be, and maybe just having an interest, and like you say, like you have a funding mechanism for some of the summer work, that could be quite important, because I remember, living off of like $500 a month, in Boston while doing some of this, kind of summer work in the lab there. And, even shunning other jobs that would have, probably been more, that would have probably paid better, but, essentially just wanting to get back into the lab and do that, even if it was sort of, relying somewhat on, kind of still input from parents and things to be able to afford it. But, at any rate, I mean, I think, some mixture between, if the model that you're driving could also connect up with some, if it's in the same university, like an ability of the student to basically come back and kind of continue things if there was a strong interest, and might set the stage for someone then from this undergraduate stage really developing almost kind of like a master's like project, and then being able to segue into a PhD without too much additional lab training or experience. So
almost
anyway,
like a long-term internship, almost sort of a long thin internship throughout that period. So how have you found living in the UK? So outside of the science, how have you found that side? Has that been very different? Was it unnerving actually? Oh, come on. This was 10 years ago.
We've had very different, kind of like I grew up in the Hudson Valley, in a town called Poughkeepsie, which is also kind of a college town. It's a little bit, it reminds me quite a bit of Oxford, because it's kind of a medium-sized city, industrial city. Like they had a lot of IBM, basically, so kind of computer manufacturing and things and design. So a lot of technology oriented things, but also Vassar colleges there. So it was kind of, it's sort of, and then surrounded by farms and, kind of countryside and, of course, other, the, Catskill Mountains and things like that. So it was kind of like this sort of nice mix of sort of like rural and city. And, but then we went to Boston, St. Louis, New York City, lived in Manhattan for 10 years, 13 years actually, before moving to Oxford. So going from Manhattan to Oxford was quite a change in lifestyle, no matter how you slice it or things. Yeah. I mean, kind of like a unique urban experience to a much more, kind of, again, like this sort of medium-sized city with very heavily influenced by a college. So, but, we have great neighbors here. So we have a house in North Oxford, and we kind of, our neighbors are really great. So yes, one of them was academic. One of them was, a former Oxford chair, and one of them was, basically just people like a guy who had a garden, built business and things. But and just wonderful people, and so enjoyed those interactions and others and also just it's a very international community. So like our institute, I would say, is less than half of the PIs are probably were originally based in the UK. They've really come from all over. So we don't really have much, feeling of there's a kind of a transition, I suppose, between, the being a fully expat, like, and not having these connections to you get this sort of very familiar aspects of international science, which we also had at other universities we worked at. So again, like so it's not such a shocking thing, but I mean, some things are just kind of different about kind of, but I think in a charming, pleasant way. I think particularly for the say Oxford area.
And so you said it was your daughter you said that went to university?
Yeah. So our daughter. Is back in the U.S. still, and is actually kind of returning to school now. So she, didn't finish college. She was, when she reached college age, she decided she, didn't want to do, university at that point or go to college. She, basically has an interest in kind of veterinary, very bright kid, veterinary, was working at that point in kind of a vet tech kind of direction in New York. And then she, kind of met the love of her life as who's a pathologist, and basically then started going around with his training there in Kansas City, New Hampshire, then Pittsburgh for a while, and now they're settled outside Washington, D.C., which is actually quite convenient because I visit Washington a lot and for various things, and that gives us an opportunity to see her more. But essentially, so she's going back to school now, kind of in a biology area, probably maybe with an interest in long-term interest in medical school or veterinary pursuits. But that would be that's something that she's doing, maybe what one might call as a mature student in the UK.
So I was going to ask, sort of, how often do you actually get back to see her? Because that's the other side. There's one thing when a child fledges the nest, but actually still want to kind of see them and be close. And of course,
yeah. So this was why when you look back on things, you say, it's so easy to get back and forth between the US and the UK. And, we had my lab was maybe initially 50-50 from like 2013 to 2015 or so. So we're going back and forth like all the time in New York, and she was in New York. So yeah, again, we were seeing her all the time. But then, with like the pandemic, in 2020, that, it kind of changed that whole thing. And also that was, so this was when her and her husband, Ryan, were kind of, bouncing around to all these different places. So we never visited them in Kansas City. We never visited them in New Hampshire because it happened in this time frame where there was just very little travel. And now that when they were, we did get to visit when they were in Pittsburgh, and kind of now catch up pretty regularly that they're in the DC area. It's quite easy. So again, it was there are all kinds of things we didn't anticipate, like Brexit. Trump, maybe Trump 2. God, this is like, all these things that are sort of, so it was good that we left. Is it, bad that we left? So the world's a surprising place. And I guess, but, we roll with these things, and we used Zoom a lot, basically for our communication during the pandemic, and to stay in touch.
So actually thinking of the world changing and everything else, going back into the research side of things, funding obviously is changing, always evolving, and the funding arena is always changing. And over in the UK, obviously, it's very competitive. You have to write your grant proposals to get your funding. So I believe, were you Wellcome Trust funded to start? Were you still Wellcome Trust funded?
Yeah. So the our original recruitment was with a Wellcome Trust Principal Research Fellowship. So it was which was like an amazing opportunity, and that really, was 7-8 years of the transition. It was really kind of like the startup package for transition in the lab from NYU to Oxford, along with support from. So the Kennedy Institute is a research institute within Oxford. It moved from Imperial College, and where it was affiliated with Imperial College in London, to Oxford in a new research building in 2013, and that's when we arrived. So the Kennedy Trust for Rheumatology Research is a charity that basically was a beneficiary of patent royalties from patents related to tumor necrosis factor-based targeted therapies that were kind of developed. I mean, interestingly, like the antibody that was the original anti-TNF that was used in the clinical trials that were run by the Kennedy Institute was made by Jan Vilcek at NYU. So when I was at NYU in the 2000s, there was this huge windfall of money from the licensing of that antibody, and the patents that were generated around kind of dealing with the anti-drug antibody effects of administering antibodies as a long-term therapy, generated lots of revenues for the essentially Imperial when the Kennedy was there, and the Kennedy Trust for Rheumatology Research, which was kind of the charity that supported the Kennedy Institute from the 1970s. So kind of had been investing in that for long term, and the institute is kind of a reinvestment of those funds. So it's kind of so from a funding standpoint, the Kennedy Institute is a long-term project of the Kennedy Trust for Rheumatology Research, and we benefit from that quite a bit. And but it's it's basically they're a funder. We write grants, and this is similar with the Wellcome Trust. And I have to say, I guess I think in the U.S. I would say the NIH pay lines and things fluctuate over time. But otherwise, the system is very stable. Like the funding, platform has really, maybe the biggest thing that would have changed in my lifetime maybe was the emergence of the Howard Hughes Medical Institute as a major funder. And I guess at the same time, I guess like the Wellcome Trust became a major funder in the UK, but is a bigger percentage of UK funding than, say, HHMI or something in the US. So clearly, when the Wellcome Trust changes its mind about something, that has a huge impact on research in the UK, and it really forces like lots of people to recalibrate and kind of imagine things. So I think I feel like the funding landscape in the UK is. And also, with the ERC, like our relationship with the ERC, we've benefited from significant ERC funding. Also, we had an advanced grant and have a synergy grant now, and even navigating the Brexit process created quite a bit of volatility and uncertainty. But again, it seemed like the best bet at that point was just to keep writing those grants, to keep submitting grants to that organization. And there have been different ways that they've been funded over time, over the past few years, but they've always been funded if they were competitive in that system. So I think the bottom line is you just keep writing grants, and as long as you're happy with that, in that process of thinking about things and writing and discussing with people, and that's part of the job in some ways. It's not just a way to get funding, but it's also part of the process that you use to basically innovate, and you're challenged in some ways to innovate and keep moving forward. That as long as you can kind of be at peace with that, then I think it's actually a healthy system.
So I should just I just realised my pen is actually from Howard Hughes. It's from Janelia. So
it's right. And I think and certainly HHMI has been a model for things that, say, Wellcome has done over time, and but I think I'd say it seems as an organization like Wellcome, maybe every 10, 15 years or something, asks, okay, is what we're doing the right thing? And then basically shakes things up. And I guess you could say in some ways, in terms of like you see some costs in terms of some continuity, and say individual organizations being able to maintain certain kind of skills and things may be challenged a bit. But I mean, in general, I think the scientists are adaptable by nature. Maybe is one of the phenotypes that we select for, and you kind of you work with it.
I think the most successful academics are very good at moving and adapting. It's a critical aspect, I think, in academia to be able to adapt and evolve with the funding models, which can feel instinctively wrong for some because they're very naturally keen, or their passion is one area, and sometimes you have to evolve that area to fit how the funding's going. So I actually have a different question. How often do your grant proposals fail?
Probably over time, like our eventual success rate with revisions is probably okay. Might be more than 50%. But individually, in terms of individual grants and whether they get funded or not, in a way, you might almost say, for my NIH period and other things, that it may almost be closer to almost like what you might say like random chance, like closer to the pay line. Because I guess early on, I did struggle to get my first NIH funding when I was just starting my lab in WashU, and so I guess one important thing was that the Emily Nanaway, my chair at that point, and Steve Teitelbaum, who was kind of like I was kind of sitting between two departments, and they were the heads of those departments, and they supported us. They sort of gave us, they put us under some pressure, but they did support what I was trying to do in my group, and saw that there was something there. And so we went from getting like the idea that we were pitching, maybe we weren't quite communicating it properly or whatever, but when it was an idea and we were kind of building up the underpinning kind of technical elements of it, we were getting like triage, which would mean in the NIH system, US system, would be lower 50%, and then we went from almost like that kind of triage point to getting like the top score in a grant round when it worked. So that was sort of, and again, like people, I've talked, kind of just informally spoken with people who, saw this development, from say like the study section side or something. I'm not supposed to talk about these things, but, again, it was like many years later, and kind of just sort of in a general way saying, wow, that was really interesting to see that, because, they were trying to decide, like, is this something that, we can support or we should support, or it looks interesting, but it's, we don't quite get it. And then, eventually saying, oh, that's what they were trying to do. And then realizing, okay, that's actually okay. So holding on to that point was a bit of a, there's a bit of a cliff edge there, but it did eventually connect.
If you never got that funding, well, it's career making, isn't it? And to have got those results, which I guess I'm presuming is around the supramolecular structures of the synapse, immune synapse.
Yeah, right, exactly. So this was this kind of, paper that was published in Science in 1999. And when that was, when that data, even before the paper was published, when that data went into the grant. So there's always like this kind of grantsmanship thing about between preliminary data, which isn't published yet, and the data that's published, which you've already done, and therefore you can't write for funding for. And so I believe we had that data in the grant while we were like in revision for the paper, and basically the paper was both well reviewed, and at that point that work in the grant was seen like, oh, okay, that's what they're trying to do. So it was kind of so it was our sort of so it was a kind of like a eureka moment that was actually shared with all of these kind of peers basically who we all know each other and things, and it's sort of just they were just trying to understand what I was going on about with these supported bilayers and things, and where was this going to go? Was it just sort of like a niche biophysical tool, or was it something that would actually impact the way people think about these things? So
it's interesting you said about it. You talk amongst your peers, and your peers were judging whether it was. So that suggests you were quite well networked, even in an early stage of your career. So how important would you say networking is, and what is the best way to network today?
So I guess as a student, so I worked with a kind of a guy who was an assistant professor. So it was really, his lab was in an early stage, but he was clearly on an upward trend. And again, like for us, he had a transformative technology. And so as Tim Springer, so as a student, he worked with Jack Strominger. So again, so he was like well connected in the, say, power structure of, immunology in the 1970s and 80s. So clearly working with, a very, very highly well-known scientist, and his work was, outstanding work as a student. But then he did a postdoc with Cesar Milstein in the UK. So again, and that's kind of, in a way, one of my early connections with the UK was that, these people that I was, working with early on did a lot of their, say, postdoctoral research in Europe at that time, and, working with these, giants of, in various fields. And, he learned how to make monoclonal antibodies in Milstein's lab, and then came back to Harvard and basically started making antibodies to things, and made monoclonal antibodies to a number of adhesion molecules used by lymphocytes for cell-cell communication. And, the problem that it was in front of the groups that I was working with as an undergraduate was purifying membrane proteins, like receptors that were involved in cell-cell communication, was a huge challenge. And just being able to do conventional biochemical purification was really impossible, just too expensive, and would never work because of the physical heterogeneity of the molecules. But these monoclonal antibodies, one step, millionfold purification. So when I was a student and basically started playing with that as just doing a lab rotation with him, it was like, oh my God, they've solved this huge problem that my undergraduate lab was basically destroyed by in some ways. Really, like, I guess they got grants to do this, and then they couldn't do it because of the challenges of purifying these proteins and these like detergent extracts and things. So again, it was like, so the networking thing came in from getting into an area that was kind of emerging, and then at least for me, then basically having, early papers in that. So even as at the beginning of my graduate work, say I started in, 84, 85, had a paper in 1986, again, using these monoclonal antibodies to characterize, one of these adhesion molecules, one of many, now, but maybe an early one at that point. And then just, starting to go to meetings and just meeting people through, kind of emerging collaborations. Like these antibodies were great collaboration tools because you could kind of distribute them to people. And this was sort of like in immunology, there were these workshops that they had starting in the 1980s, where people realizing that they had these antibodies, and they were all naming the molecules they recognized, and just kind of profusion of ways. So one molecule might have 20 names based on different people making these antibodies. So they decided to get together all of the people who were making antibodies and analyzing these surface molecules, exchange the antibodies, and this started the CD thing, which some, CD1 through now they're into the hundreds. And this was like an early, kind of like informatics project. It was like the Genome Project. It was like the Cell Atlas. It was basically a collective thing where people got together, pooled their results, used a clustering framework to basically identify the different antigens, and then published these in these big books, which you can still probably find in libraries and things. And I think there's even some online access. But again, this was like a huge formative thing. And those created enormous networks. So we got lots of, we would get antibodies in, because Tim was an antibody producer, he would get these antibodies in. So we had like hundreds of little tubes of, kind of these, highly concentrated antibodies. And we would just, do all these experiments. And then we go to these, meetings and argue about who should be CD1 or whatever. That was like when in one of the initial meetings, they had to decide, okay, what would be CD1, and this was sort of, somewhat of a political thing, but it was also somewhat of a collective, form, sort of process. So again, I thought that was, so in my career, that was a networking opportunity. So I suppose, these things are, now going on and probably on an industrial scale. And I think, getting involved with those kinds of things is a great way to network, in addition to, filtering all of the social media type stuff. But, I think certainly getting together with people who are doing things and have relevant, tools and stuff, and trading those, like even microscopy, say, one can, certainly network a lot around microscopy technology, because different places have different, strengths and things, and you can, not everyone can have everything. So that can certainly be a way to, connect up with people and say, hey, I've got an experiment that would be great on your microscope.
So I had a question, which this leads really quite nicely onto how competitive would you say the field is? Is it a very collegiate field, or are there different parties? Is it nicely competitive, or is there some aggressive competitiveness in your area at the moment?
Yeah. Well, it's interesting because we've worked on, let's say, T-cell antigen receptors for a long time. And one thing you run into in any field is, at any particular moment in time, people will have kind of like a working model, and they believe that they fairly well understand what's going to happen when you induce a perturbation. So when we were looking at these this kind of immune synapse concept, and this would involve T cells going from a motile state to stopping, and so we had two. It was interesting, when you then kind of just query everyone, what do you think will happen when a T cell sees antigen, when it sees and it recognizes its proper MHC peptide complex? Some people were perfectly happy with what we were seeing, which is that they stopped, but other people were thinking, no, they have to keep moving. And then there was some in vitro data that would support that. So it was kind of, interesting, like around 1999, 2000, that time frame, you could have a pretty, robust debate about basically which one is which. And nowadays, there's still a lot of, say, for Oxford, like one of these, say, Oxford-based theories for T-cell receptor triggering would be based on movement of CD45, a large phosphatase, away from the T-cell receptor through some, the membranes come close together. This protein with a large extracellular domain gets pushed out, and that creates an environment where the signaling can basically go forward because the phosphatase is dephosphorylating, and the kinases then can have a chance to, basically build up a signaling complex and condensates and all these kind of things. So there's sort of like a big argument about conformational dynamics of the T cell receptor right now. And it's kind of interesting because even as the technology advances and we get better views of what's going on, there's still a controversy. So you think eventually this has to be solved. People have to basically be able to say, okay, this is is this like a GPCR that undergoes critical conformational changes, or is it like this rigid body that just sets up this short space and then presents these cytoplasmic domains for phosphorylation without actually otherwise changing? And that is still a quite robust argument. And again, people will declare victory, one side or other will declare victory in a certain moment in time based on some new observation, like say the cryo-EM of the T cell receptor was one such event. And then the finding that if you do cryo-EM on T cell receptors in detergent micelles, they look exactly the same, like different receptors look exactly the same with or without ligation. But then, if you do, now it's starting to emerge, and there's a preprint out that basically says that if you do the same structure in a membrane disc with a bilayer, that now it looks like it can actually, exist in a number of states, and that makes the quality of the data much lower. But it actually, because it's you're basically trying to catch a falling structure of some kind, or you're looking at something that's actually breathing. But I think again, it continues to be like a hot topic, and we're going to have a meeting in June where we'll have a chance to see some data from different perspectives and continue to argue about this.
Which is why I guess science remains healthy. Mike, I'm going to change tack and ask you some quick fire questions. Okay. So are you an early bird or night owl?
Night owl.
Night owl? PC or Mac?
Mac. Back from 84. So I was hooked in 84.
McDonald's or Burger King?
Neither.
What would you choose if you had to choose? What would you choose outside of that for your fast food?
I love the like sort of falafel stands, little food trucks that have like the the lamb and the sort of Mideastern fare, kind of quite probably equally I should probably say equally unhealthy, but basically a little bit more like a mom and pop sort of level. Yeah.
Coffee or tea?
Coffee. Even now I've been in England, it's afternoon tea is still not a normal thing.
I'll keep you with coffee. Beer or wine?
Sorry,
beer or wine?
Oh, either. I would say beer.
Okay.
If I had to choose one, and there was no social pressure. Like sometimes at a dinner, there's a collective decision, like, okay, you go with a bottle of wine or beer, and then I can go either way in that setting. If a guest wants to do wine, that's great. Maybe more towards red wine, but basically if it's just sort of like everyone's doing their own thing, I might go with like the Belgian ale or something. Yeah.
Okay. Oh, that answers the next bit if it's a Belgian ale. Chocolate or cheese?
For dessert, probably more towards chocolate.
Okay. UK or US?
Oh my. So, I just say UK at the moment. We're here. We're all in.
You still got a US passport? I'm just checking. You still got a US passport?
Yeah. Right. It's hard to get rid of that. It's actually it's like a leech.
TV or book?
My wife is more of a book person. I'm probably more of a TV person. Yeah.
And what are you watching?
Or visual, like no longer TV. It's more like your computer screen. Sorry.
What are you watching at the moment?
We've been doing the three-body problem. I think we just completed the current set of the three-body problem, which is basically, so it's kind of, I guess I use these, kind of services quite a bit, but that's a Netflix thing, I guess. But so streaming services, and So we kind of not quite binge watched it, but basically took it in over a period of a few weeks. And that's a sort of, I've read those books. So I guess I did read the books first, but I was interested to see how they would basically present it. And certainly it's easier to present something with that kind of scope as a series as opposed to just a single movie.
So is there any TV or streaming that you'd actually, that you're going to confess to, even though you don't really want to confess to watching? Anything really trashy that you enjoy watching?
I've done all the John Wick movies.
That's not trash.
That's probably that's actually that's mainstream. Yeah. Well, and maybe the equivalent kind of shoot 'em up sort of trash. Obviously that's the high culture version of it, but there's also I'll some of the streaming when I just want to unwind would be in some cases this kind of along those lines.
Which leads nicely on to, what is your favorite film?
Oh, like a single favorite film. It's based on things that I've watched a lot of times. I guess, I really do like the Christopher Nolan films, like Inception, and I guess and there's one now that I'm actually going to blank on the name, but it was basically one of the earlier films with this sort of reverse chronology. I'm just blanking on the name of it, but basically it was something to do with memory, like Memento or something like that. But basically it was essentially so probably I've kind of enjoyed some of those things that kind of play with your mind a little bit in terms of thinking about kind of dreams or time, or and again, like that was the thing where it was kind of someone who has short-term memory loss, and kind of like how that could be, how that would change their perception when they basically reset after having forgotten everything that's happened in their immediate past, but they have a long-term memory of their distant past. So it's quite a again, you have maybe some conundrums around that.
And what's your favorite food?
Probably from a family kind of traditional standpoint, like the Maine lobster is sort of like would be sort of like one of the Dustin sort of kind of favorites that I'll still rarely, very occasionally, but kind of often sometimes take advantage of.
So at home, are you the cook or the washer-upper?
Usually the washer-upper, but there are a couple things I do, for dinners that are sort of that rotate into the. But usually I'm the. So during the lockdown periods and pandemic and things, we did kind of like diversify our repertoire of things that we were, kind of preparing, and we're still kind of in that, working through the things, or now kind of repeating some of the things that we made in that time frame for the first time.
Okay, I didn't ask you about when we were doing the films, Star Trek or Star Wars?
Early on, Star Trek. We. I was kind of raised on that, I would say, in some ways. Like that was something that was, again, with all of its, kind of period, strangeness. So did enjoy that.
Okay, X, Twitter, or LinkedIn?
So, LinkedIn has been the most useful for me because, it's a great way to get in touch with people and companies that don't have good websites, or at least websites that let you really track people in the organization. I think that's sort of like and we do a lot of collaborative work with pharma and small biotechs and things around this immune synapse topic, which is kind of in a way, sort of a framework for how some of these, drugs, immunotherapies and things work. So that's been, so LinkedIn's been really useful, like Twitter, X type thing. I have an account and I'll tweet papers, like, preprints and things, but basically most of the real utility comes from LinkedIn and connections made there.
Okay, and final quickfire question. favorite color?
Green. As a child, I had kind of I would say like a hyperactivity, and I was a slow to learn things. And, green was the first color I could really nail down, as again, when I was really young. So that I kind of stuck with that as a favorite color.
That's the best answer anyone's given to date. Although looking at your shirt, I'm surprised you didn't say green fluorescent protein or yellow fluorescent protein.
And it's green fluorescent protein. So that reinforced my decision, that basically this is the evolved, fluorescent color that you use to. But of course, you're switching blue light, so it's all the whole spectrum basically is now of, it's really. I'd say the visible spectrum is my favorite part of the spectrum. Although we're using X-rays now too, and electrons and stuff. So I guess it's.
So taking it, you mentioned when you hyperactive learning gets sort of green, the first color you can remember doing. What was the first job that you can remember ever wanting to do as a child?
It's interesting. Like I really, I guess if I were to filter it and kind of, present it, I really kind of love the idea of being like a naturalist studying nature. So that was sort of, I remember actually saying this, I want to be like a hermit, because that's what I thought, this was sort of like, if you're watching these, the studying animals in the, in their environment, could be in a desert. I was quite interested in desert. That this would be, sort of like being also the whole lifestyle would be sort of like, you're camping. It's sort of, it would be sort of like, but that was my first interest. And I actually kind of, even when I got to college age, I did actually like look at like wildlife biology programs. I applied to one at UVM, and that was, again, kind of before I decided to just go with biology at BU. And again, BU was kind of like, okay, it was like, Boston, versus Burlington, Vermont. I was kind of, maybe more drawn to Boston as a student at that point.
So, and from the extreme of what you wants to be, if you could do any job in the world for a day or a week, just to know what it feels like, what sort of job would you like to do?
Oh, that's a good question. Maybe at this point, like like a CEO of a big company, just heading a big company or something, and just seeing what that, I don't know. On a given day, nothing may happen, but it may be, I guess, on a busy day, like a busy day for someone who's running something really large. Because again, I don't have any idea how anyone would view that when I have a hard time managing, like a lab with 10, 12 people or something. How do you deal with this kind of huge hierarchy? I suppose it's a lot of delegation, but I guess it could be. And again, we're like I said, I mentioned earlier that we're kind of spinning out a company, and right now I'm just like kind of like an academic founder, so I'm not going to join the company any, and I have there's a student from my lab and another Oxford student who are basically the CEO and the CSO. So they're starting this, it's just like that little thing at this point called Brands of Bio. But I'm really excited about that, and again, like I'm kind of interested to see, because they've been kind of, it's interesting, like this generation. I don't know if it's this generation or maybe it's always been present, but these guys have had lots of entrepreneurial training as part of their, Ashwin J. Narayanan, the guy who's one of the guys from my lab who's starting this, has done lots of these like kind of pitching competitions and all of these sort of entrepreneurial sort of, just workshopy kind of things and educational experiences that he's done in, parallel with his DPhil and his undergraduate work and things. So he's been kind of building towards this for a long time. But I think it's like, and again, they're doing it with a incubator called Y Combinator, or accelerator, I guess you might call it, called Y Combinator. It's out in San Francisco, and they're getting lots of, kind of just huge learning curve of basically talking to their, people there, and people from these, successful startups of the, past 10 years or something. So it's really, again, it's quite exciting.
It's a brave thing for them to do.
Yes, they're all in. And that's, so at least, the Y Combinator's thing is like the success of this company is, we've had some input into some, kind of observations or phenomena that they're, building on, but really it's going to be them, who will make it or break it, because that's the, they're putting their full time into that. And, again, I think it's exciting to watch. And again, this is something that as I've gotten, even from, my first move from St. Louis to NYU, being able to be involved in supporting someone who's just starting out is also like a really nice thing to be able to do once you've gotten through that sort of threshold of getting to the next step or something. Like, you've gone from, you've got tenure or whatever. And when we moved to NYU, there was a junior PI named Wen Biao-gan who was interested in like having 2-photon microscopy, but didn't have sufficient funding as a assistant professor just starting his lab to do that. He was using, various things they could do with conventional fluorescence microscopy on very thin specimens, but he wanted to go into the cortex. So, when I was talking to him during my visits to NYU, he was like, I can build the microscope that's needed for this, and just from parts for it's a lot of money, but relatively speaking, less than a commercial system. And so part of my thinking in this was like, okay, we want to learn this technology. Here's a guy who has the skill set to build it, I was convinced, and has his own set of questions. So he has his own set of biology he wants to study. So there's no competition at all, and we can learn from him. So that was one of the attractive things about moving. And we kind of, with our startup package, we had to bankroll the microscope, and he had a paper in Nature within a year. It was amazing because he knew he wanted to basically look at dendritic spines in the cortex, and he had an idea about like what he would find in an adult animal, and it was and he was, again, they went in and did these heroic experiments and collected this data, and he published those results, which were actually kind of going against the trend of the time in terms of like synapse stability in the brain, because he was looking at adults. Everyone else was looking at developmental systems. They wanted to see dynamics, so that's what they saw. He wanted to see, okay, what's a stable network look like that's actually functioning? And he found lots of stability. So I guess so that I guess the thing about and that so we weren't involved in those papers at all. But it was great to be able to have input in it, and we did do some experiments together, and that was a lot of fun too.
We try. It was interesting because actually, if you now go into sort of today's sort of world, access to instrumentation of whatever nature is a very big pull for academics, so they can start answer questions that they may not be able to address elsewhere if that technology isn't available. Multiphoton back then was obviously a niche emerging
rare thing.
Microscopy at that point. Have there been any really challenging times, difficult times in your career that you've had to overcome?
Well, I mean, really just, that first, in a way, like we had a lot of fun in the lab. Like I was actually working in the lab for the first 5-6 years. And maybe that was even like a problem. I wasn't working hard enough on the communicating the information in the grand small lab, but that was great. And but there were stressful periods there and things where we like, okay, this is not working out. Like we need to change something. And again, there was support and there was pressure from the leadership in our department. So I think so that was tough in some ways, but a lot of fun in others. So
what would you say has been the most fun? What if you could repeat a year? What was the most fun year that you've ever had? It's going to be the same time, isn't it? I can see this coming.
So that working on that paper was a good time, but I'll pick a different thing. So there was in 2015. So after we moved to Oxford, we got invited by Ron Vale and Mike Rosen to do a summer in Woods Hole. And it was sort of like maybe 2 months, and they had an HHMI a collaborative workshop grant of some kind. It was basically like, to basically bring together people from like 3 or 4 labs. You'll have this, set up this little sort of lab in Woods Hole, which reminded me of the labs I worked in as an undergrad at BU in the 80s, with actually similar level of regulation. I think it's become more, normal now, but at least at that point in 2015, it was kind of like, very informal in a way, in a regulatory framework. But it was sort of, I actually got to do some experiments and work with some of the things that people, I'd worked up to maybe 2000 in the lab. So for 15 years, I was doing nothing in the lab. And I got, like for a month or so to basically just tinker around in the lab with a postdoc and a student who joined there. And then all the other, postdocs and students from, Ron's lab and Mike's lab. And these are now like all colleagues, in the field. They're all people who've, are still in the mix in some way. As I was just visiting Marcus Taylor in Berlin just last week, and kind of hanging out and seeing how he's doing at this Max Planck Institute where he's working. And he was one of the postdocs who came over with, I guess, like Future Mayor, and he had maybe connection with Ron Vale too. So it was sort of like, it was a great little, just sort of let's do things together around condensates and signaling. And they had like, 10 microscopes or something, and everyone just sort of, did experiments, some with some desire to basically trade, our little trade secrets and things that are sort of, you can't really even write enough detail in a protocol in a paper to describe these things, but we all got to, kind of exchange them. So it was really fun. So you can't do it every year probably, but some people do. I mean, some people pilgrimage back to Woods Hole and just tinker around in the lab. But for us,
sounds more focused, given that collegiality, networking, but those sort of tips, the tricks of the trade type tips, and sharing those in an informal way. That sounds a very smart idea, actually.
Yeah, and this was like all, maybe several labs in this area in different fields of biology, like not just immunology, which was our kind of interest, but people working with other types of cells and stromal cells, and just sort of it was kind of a bit of a melding pot, or kind of a chance to just sort of cross-pollinate, and again, it was great. It was a lot of fun for me just because I got to tinker around in the lab a bit, and it was sort of like I felt like I wasn't being irresponsible. I was kind of it was part of it was a worthwhile effort, that I could have that reconnect sort of.
Out of work, what sort of hobbies do you have? I know you got a pet dog, Potter, I think.
Yeah, so we've had various dogs over the years, starting from when we were kind of just going off to our postdocs, finishing graduate school and going to postdocs. So we've always had the dog training and keeping the dog kind of sort of like was our first, in a way, like our first child, got us ready for having a family with our daughter, and then kind of now kind of is our again our little boy at this point. But the I mean photography, I mean, it may sound a little bit too close to what I do in some ways, but from high school had an interest in maybe teenager years, had an interest in photography with film, now mostly digital, obviously, but I'm still carrying around an enlarger. So, one of these days I could go back to film if I found a good darkroom to set up, a place to set up a darkroom. And of course, labs don't have those anymore because everything is pretty digital. I think we still have the film developer, maybe, because some people are still hanging on to ECL, but it's, or this luminescence-based gel technology. But, the opportunities to maybe at some point I'll see if I could rent some space in a darkroom and play around with that. That would be fun.
Yeah, I when I started at York, we had a couple of dark rooms. One's now got the multiphoton microscope in it. Good place to put into a dark room. It's an ideal place.
No windows, yeah.
And the other one's now got a label-free base focus system in there. Both of them gone as the electron microscopes really went digital at that point.
Yeah, sure. Right. Film became obsolete.
So your hobbies are Phil. It's actually my PhD supervisor, Richard Cherry. When he retired, got into photography in a big way, and then started to get quite competitive in his club and everything else. I guess that eye for imaging makes a
Yeah.
big.
Well, nature, I remember I mentioned that, I was interested in nature early on. And so photography, photographing, we've done at least, we did one big trip to Panama where we did sort of like bird watching and photographing, lots of photography of, all these, beautiful birds and things. So, I could see, if we had more time, maybe retirement or something, kind of, getting into that more, as sort of like just sort of it's not so close to the microscope work, and it gets you outside.
So we are up to the hour already, but I have to ask you, have you got any regrets? If you go back and change something, is there anything you would change?
I think mostly around just, kind of communicating better in some situations. I guess, I think like in academic settings, I guess what I learned through several transitions and things is that, being, completely open about those kinds of, just basically what you're thinking at any time in terms of, uh, if you feel like you need to make a change, to just, let everyone know that that's what's happening. Because I'd say, like, when I left WashU, I kind of regret not communicating more clearly with my chair at that point. And then when I decided, when the kind of the idea of moving from NYU to Oxford came up, I was much more communicative about that whole process and what my thinking was, and it was obviously much better. So I think, like, just, I guess, as, sort of like communities, like, again, you think about, collaboration, all the peer review stuff, that we're really completely dependent upon communication with our colleagues, and of course, like the closest colleagues, like the people in your department and things, to basically to be, really open with them about what those kinds of things, like, things that would affect, kind of big things like moods and stuff like that, not to be, because I was kind of, I guess at that point, maybe I was a little bit, maybe just a little bit lack confidence to basically just say, hey, I'm thinking about this. And it turned out that they knew everything, so everyone knew everything that was going on because they obviously have their own networks and things. So it was just kind of, in the end, I think that kind of open communication is sort of a key, and again, I think, it's possible that in other job settings that maybe there are even like kind of confidentiality issues or something that would say, that, in which case that might be different. But I think in academic pursuits, I think we're, really this kind of, my experience, openness would be the way to go.
And on the theme of communication, although we're just over the hour, I should just point out for everyone watching, listening, that you actually do quite a few different YouTube clips really explaining the immunological side.
Oh yeah. Well, this was at that Woods Hole I mentioned in 2015. So Ron Vale and his wife were basically organizing this iBiology series, which is, has lots of fantastic, these kind of videos, but they did these kind of like weatherman type things, like where you have the green screen and you kind of redesign your slide so that, 1/3 of the slide is blank that you can stand in basically. And then, that you're basically pointing at the, rather than using a pointer, you are literally in the picture pointing at whatever you're trying to communicate. So we made the series of 3 videos that were kind of, maybe a 2015 snapshot of where that field was, but kind of, trying to start with more basic immunology concepts. And I guess like the first one is probably pretty accessible for like a science, like a high school science student or something on that level. Like maybe not like a total, someone who's, no longer, thinking about science at all. But I think, again, that was a lot of fun. And even just kind of preparing that and kind of, getting into the mindset to kind of, do that was a lot of fun. And that was part of that thing. Like, so I could do experiments and we did those videos in that same 2 months.
So the impacts on that sound really good. We're well over the hour. We have to stop. And there's so much I wanted to talk about. We haven't talked about spatial imaging. We haven't talked about the first microscopes and where you see the future going. But we have to wrap up. And Mike, it's been a real pleasure. I hope everyone listening, I think there's been so much useful advice in there that I think we can all learn from as well. And just carry on inspiring. Mike, thank you very much.
Yeah, thanks, Pete. It's a pleasure. And happy to catch up on some of these other things at some other point.
Thank you for listening to The Microscopists, a bite-sized bio podcast sponsored by Zeiss Microscopy. To view all audio and video recordings from this series, please visit bitesizebio.com/the-microscopists.